This is the second video of biotechnology, and in this video we will be discussing about endonucleases, alkaline phosphatase, and ligase. Okay, endonucleases, as the name suggests, it cuts the uh, segments of genetic material. Okay, and what was discovered? This was discovered. Okay, around 1963. Okay, that this endonucleases are also known as molecular scissors. Okay, or it is also known as restriction endonucleases. Okay, why and how? Restriction endonucleases, because we will discuss it about it a bit later. Molecular scissors, because it cuts, it rises. Okay, now it was discovered in 1963 that in prokaryotes there is a defense mechanism. Okay, the defense mechanism consists of two activities. One is the methylating agent, and another thing is endonuclease activity. Okay, now let's consider a E. coli, the cell of a E. coli, an E. coli. Sorry, for example, this is E. coli. Okay, and here bacteriophage virus comes and binds. Okay, this is the genetic material of E. coli, as we have discussed in the previous video that no nuclear envelope is there, so I have not mentioned any nuclear membrane here. And for example, this virus has injected its own genetic material here. Okay, now this endonuclease will come and it has some restriction sites. Restriction sites means the sites where the endonuclease is specific for that sequence. For example, here there is a T. Okay, and here also there is adenine and thymine. Okay, now this endonuclease, for example, endonuclease X, the the restriction site for endonuclease X is AT sequence. So this X will come and it will lyse this AT sequence, and it will also lyse this AT sequence. If both the sequences are lysed, so it will be cut and this too will be cut. If it gets cut, if it gets split into two parts, will it be functional? No, it will be denatured. So to prevent this kind of suicidal activity, there is a thing that we discussed, methylating agent. So what happens is that this methylating agent methylates one of the two bases, either adenine or thymine. As a result, even though the endonuclease named X, okay, its restriction site is AT, but because there is a change in the proximity of the sequence, so it will recognize it and it will leave it, and it will go and lyse here. So if it lyses here, it will cut into two different parts, and if it gets cut into two different parts, it will split, and if it splits, then the foreign DNA is denatured, right? So this is the defense mechanism, and because it is denaturing the foreign DNA and it is restricting it from taking control of the cell of the E. coli, so it is also known as restriction endonucleases. Okay, I hope it is clear. Okay, so now we will discuss about some more things. We would discuss about alkaline phosphatase. If this is the sequence, okay, here there is OH and here there is phosphate group. Okay, the OH is on the 3' proximity and the phosphate is on the 5' proximity. Actually, the DNA is generally double-stranded, covalently closed circular DNA. Now, to deal with it, okay, it's okay, but we need to insert a thing here. Okay, for example, we need to insert this here. So what do we need to do? We need to cut it. Okay, to cut it, we would need endonucleases. So we have cut it. Now the sequence somewhat looks like this, right? Now there is a chance that it will again recoil and it will form circular pattern. Okay, because there is a chance that this OH and this phosphate group will rejoin. Okay, you must have read this, that if this is the sugar, this is the OH, and there is another ribose sugar, here there is triphosphate bond, this is alpha, beta and gamma phosphate. This OH attacks this alpha and beta phosphate and it forms phosphodiester bond, right? So to form this phosphodiester bond, what it needs? It needs the OH group and it needs the phosphate group. What if we remove the OH group? Will it be able to bond again? No, right? So we are doing this only. This is the mechanism that is followed to ensure that the phosphodiester bond is again not to be made, so that we can work on it. So to separate this phosphate, we use an enzyme known as alkaline phosphatase. It removes the phosphate group. So we are left with something like this: 5' OH 3'. Okay. Now we add the insert. First we add ligase. Ligase joins the fragments of the segment. Thus, we add the insert we want to add. Okay. Now, this will also have phosphate here. This phosphate and this OH will again form phosphodiester bond. So, what will be its structure? Right, this is the structure. Now we have manipulated the gene. Now we need to go back to its original conformation, that was circular in nature, so that it can work properly. So what will we do? We will add phosphorylases and phosphates so that it becomes something like this. So I hope I am clear, right? So this was double circular DNA. Okay, we added endonuclease and we cut one of the part, then it became linear, and then to ensure that it remains linear, we cut out the phosphate group here by using alkaline phosphatase. Now after that, after we have cut out this phosphate so that it cannot again make phosphodiester bond with the OH and it will regain its original conformation, to ensure it does not happen, so we did it. Then after that we added ligase and we added the insert that needs to be inserted. Okay, so it looked something like this. Okay, after that we added phosphorylases and phosphates, okay, to ensure the 5 dash end is also joined with a phosphate, and then this OH and this phosphate that is joined because of this phosphorylases and phosphatase. These two form phosphodiester bond, and it formed again the circular fragment of the gene. Okay, the circular gene. So this was the mechanism by which the alkaline phosphatase works. In the next video, we will see some more things about endonucleases.